Good morning. On behalf of Department of General Pathology, we are continuing with our e-content learning lecture series. Today's topic will be on Paxil volume or hematocrit, as it is otherwise referred to as. Now, Paxil volume or hematocrit is defined as the ratio of total volume of RBCs to that of whole blood. It indicates the relative volume of RBCs and plasma. Example, in conditions like anemia where the RBC levels are much reduced, correspondingly the Paxil volume or hematocrit values are also simultaneously reduced. Now coming to the methods of estimation of Paxil volume. There are various methods by which we can evaluate the Paxil volume. Now the commonest method employed in the laboratories are the Winthrop method. The Winthrop method, it is comprising of the Winthrop's tube. Winthrop's tube consists of a thick glass tube. Now this glass tube measures around 11 centimeters in length and has a bore diameter of 2.5 millimeters. Now the bottom end of the tube is closed and the top end column of the tube is opened. Now the total volume capacity of the Winthrop's tube is around 1 ml. It is calibrated as double graduations. On one side they are calibrated from 0 to 10. On the other side they are calibrated from 10 to 0 in an ascending and descending fashion. Now coming to the anticoagulants, which are the most preferred and used during estimation of Paxil volume, is the EDTA. The expansion of EDTA is ethylene diamine tetraacetic acid, or otherwise we can use dry heparin or double oxalates. These are the most preferred anticoagulants. Following the anticoagulants, now we go on to the principal method which is followed in the Winthrop's method. Now the principle of this experiment is anticoagulated or whole blood is centrifuged at a speed of 3,500 rotations per minute or RPM. Here the RBCs comparatively which are heavier than the WBCs, platelets and plasma, they settle down or sediment at the bottom of the test tube, and the volume of the RBC cell mass denotes the hematocrit value. Now coming to the various methods. Now to start with the Winthrop's method. In a Winthrop's method, the blood is collected in an EDTA vial. The blood is anticoagulated blood and the blood sample is properly mixed thoroughly so that the plasma content and the solid RBC cell content of the hematopoietic cells mix properly. With the help of a Pasteur pipette, the blood is drawn from the vial and it is transferred into the Winthrop's tube. Now the tube is filled up to the top mark and slowly it is allowed to get centrifuged at 3,500 rotations per minute for a total duration of half an hour period. Now the readings of the Paxil volume are noted from the bottom as the RBCs are comparatively heavier, so they go and settle down at the bottom of the tube. Now this Paxil volume or hematocrit is expressed as a percentage value. Now once the centrifugation has occurred, the bottom layer is composed of RBCs. Once the centrifugation has occurred, the different layers are noted. The lower layer is composed of packed RBCs. The middle layer is what we call as the buffy coat, and this buffy coat is reddish gray in color, and it is the layer which is formed between the RBCs and the plasma, which is primarily composed of WBCs and platelets. Now coming to the upper layer is composed of the plasma, which is normally clear or straw colored in nature. Now, there are various conditions which are associated with the Paxil values. Now, once the Paxil values have been determined, we can calculate the red cell indices, namely the mean corpuscular volume and the mean corpuscular hemoglobin concentration. Now, coming to the normal values of the Paxils, in males, it is around 40 to 50%. In females, the normal values are between 35% to 45%. In a small infant, the Paxil volume is around 45 to 70%. Now, coming on to the other methods of Paxil volume estimation, we have two other methods. The second method is what we call as the microhematocrit method. In this method, a heparinized capillary tube is used and blood is filled by the capillary action. Now, the bottom end of the tube is sealed and the top end is open. And once again, the centrifugation of blood is done. As the tubes in this case are not graduated, in contrast to the Winthrop's tube, the PCV is measured using a PCV chart. Now, the advantages of this method is generally preferred in a newborn infant, where through the capillary blood, the blood is drawn and comparatively a smaller quantity of blood is required and the child need not be poked. Now, the method is easy, capillary blood is sufficient, less time duration, and the amount of plasma trapped in the RBC cell column is comparatively less. Thereby, it is minimizing the errors which generally occur with the various methods. So, these are the advantages of using the microhematocrit method. The disadvantage of the method is it comparatively requires an expensive centrifuger. So that is the only disadvantage with this tube. Now coming to the third method of Paxil volume estimation, we have the automated analyzer method. Now automated analyzers are the ones which are very popular in the recent days, as we can estimate large volumes of various patient samples simultaneously using this method. Now, the normal range for any Paxil volume estimation on an average is around 45%, and it is very useful in calculating the red cell indices like mean corpuscular volume, mean corpuscular hemoglobin concentration, and mean corpuscular hemoglobin content. Now, coming to the various advantages and disadvantages of the Paxils. Now, these Paxils generally are seen to be increased in certain conditions. So the Paxil volume is generally increased in diseases like polycythemia. In polycythemia, the total RBC population is greater than 6 million cells per cubic millimeter, and clinically two types of polycythemias are recognized, polycythemia vera and polycythemia rubra. The other conditions where the Paxil volume or hematocrit are increased is in severe dehydrating conditions like vomiting, diarrhea, profuse sweating, high-grade fever, and severe burns. Now, apart from this, in patients who are suffering from shock, also the Paxil volume is raised. Now, coming to conditions where Paxil volume is low, generally the Paxil volume or hematocrit is reduced in conditions like anemia and in pregnancy. So with this, we conclude today's e-content learning. Thank you, and once again, we'll meet in the next series.